Good morning, class. So, last lecture we have completed the part that is the quality control test for different containers, that is the plastic container, glass container, as well as the quality control test for closures, then rubber closures, and quality control test for the packaging material, that is the paper. Okay, so that part we have completed. Now we are moving to the next part. That part is called as good laboratory practices. It is also called as GLP. So what is GLP? GLP is an FDA regulation. It is the regulation that was created by FDA, that is the United States Food and Drug Administration, in 1978. So here they have mentioned that is the laboratory. So the laboratory here means the processes carried out for the synthesis, analysis, or the research on different drugs or different products, except lab for clinical trials. So clinical trials ही lab असते where the experiment is done on the animals. Except that all the labs where different procedure, different research is going on, that lab comes under this category. So good laboratory means the lab where the clinical trial is not taking place, where the in animals clinical labs, except that all the labs where medicines key testing or the testing of the medicine takes place. So in that lab, what are the rules, regulations, what are the ideal properties, what are the things, what are the guidelines we have to maintain, all these explained by a concept. That concept is called as good laboratory practices. Are you getting my point? So here the laboratory means all the labs except the labs where trials on animals have been done. So that is the clinical lab or the clinical trial. So except that lab, all the labs where the different investigation, different search, different analysis of drug has been done or is doing on these drugs, that lab. So in that lab, what will be the rules, what will be the regulations, what will be the different guidelines to perform all this testing, all these analysis. That's all comes under the good laboratory practices. So in the lab, that is the collection of data, execution, all these parts comes under the GLP, that is the good laboratory practices. These are these are conducted to non-clinical labs where the clinical is not done, and in the lab different testings has been done on the drugs. For that we are making some guidelines, we are making some rules and regulations. So to maintain the consistency in the result, to maintain high quality, high quality, reliability, to maintain this we require a good laboratory practices. Are you getting my point? So maintain the quality, continuity. Okay. So the purpose of this GLP is to maintain the consistency, high safety, high quality, reliability, reproducibility, non-clinical testing. So during the GLP is there to maintain our safety during the lab process. And what we are doing the testing, it should be a reliable process, it should be the reproducibility, it should give the results of the high quality. So all these things it comes under the good laboratory practices. So this GLP, the good laboratory practice, is required to understand before going for the next topics or the next things like the GMP, good manufacturing practices, or the CGMP. So before going for understanding all these things, that is the GMP and CGMP, we have to first understand what is the GLP, that is good laboratory practices. So are you getting my points? What is good laboratory practices? So here the laboratory means the labs where testing is done on the medicines. It except the clinical trials where the testing is done on the animals. Except that lab, all the lab they comes under these good laboratory practices. So during that good laboratory practices, for the efficacy, for the safety, for the continuity of result, for the reproducibility, for the betterment, we have to perform some rules and regulations. We have to go for follow the rules and regulations. So, okay, so person who is working in the lab, he should be safe. What the results what we are getting, it should be the accurate, of high quality, reproducibility. For all these things, there is need of for the making rules and regulations to make the guidelines. And this GLP is regulated and was created by the US FDA, that is the United States Food and Drug Administration, when in year 1978. So mainly it deals with the infrastructure or the facility which are provided to a person working in the lab during the laboratory work. What are the facilities we are providing? What are the infrastructure we are providing? So it should be done on the workplace. Workplace means not only the lab which is present in the college. So workplace means where you are going for the work, where you are going for the testing. That place is the workplace. So it may be the laboratory of industry, it may be the lab of the institute. So that place is called as the workplace. For the safety, for the efficacy, for the quality, what are the infrastructures, what are the facilities which are provided to the person working in the lab, that is important in case of this GLP. So I will tell you in a very simple way. Suppose you are working in the industry, suppose in the quality control lab. So in that lab, if the facility is not provided properly, okay, the instrument which is present, they are not calibrated, they have not take care of the hazardous or safety methods during the lab, proper SOP is not there. Okay, so all these facilities, if they are not provided, it is difficult to work there. It will be not safe. It will be affect the quality of the testing. Okay, are you getting my point? If you are working in the quality control, it generally deals with the testing of the different things using the instruments, and that instrument is not properly calibrated. They are not working properly. There is no SOP. Okay, safety, hazardous, all these cares are not taken. Okay, the persons working there, they have not been trained properly, and if such all these things are there, so it is difficult for there that the test will become positive, or the test will not maintain the quality. So in that case, if we are performing any analysis in that quality control lab, so there is possibility of the faulty results we will get, or we will not get the accurate results. It may be vary. Okay, so if person working there is not trained, SOP is not there, instrument is not working properly, it is not calibrated, safety hazardous methods they have not been maintained, and that case, and we are going for the testing. Okay, you have been experienced such things during the lab, our regular lab. Okay, so if you have to use the weighing machine, okay, the weighing machine is not there properly, it will not give the reading 0. Okay, sometimes you are putting the substance on the butter paper, then the weight fluctuating. Okay, and at that time, if you are taking such compounds by using that weight, will it give the proper results? No, it will not give the proper results. It will give the faulty or the wrong results. Okay, actually weight of sample is 5 gram, but due to the fluctuation, you are not understanding what will be the weight we have to take. or it will not tear properly. Of you are not using the butter paper, you are using the other things. If you don't know how to take the weight, if you are not trained properly, there is no SOP for the weighing machine, then it is difficult for you to go for the proper testing or the proper way. It is not possible for you to take the proper weight substance. So all these things can be happen in the industry if it is not working properly. In instrument it is not calibrated, or the analyst which is working there, it is not trained. If you don't know the uses or how to use the UV or how to use the HPLC, and you have placed there in the quality control lab, and you are asked to go for the testing of a sample using the UV, is it possible for you? Or you will do it, but it will not give the 100% right results. You don't know how to use the HPLC, and you have placed there, and you have asked the sample to go for the HPLC. You can do, but it will not give the proper results. There may be some changes from the, or deviations from the right results, or from the normal results. That's why the good laboratory practices are required, and this can be implemented by the US FDA in year 1978. Another thing is that is you are well trained, or the person is well trained. Okay, they have all the skills, they have studied properly, but when it enters into the quality control lab, the instrument is not calibrated properly. Okay, SOP is not there. At that time also it will difficult for the person, जरी तो train असेल, skilled person असेल, it is difficult for him to go for the to take the analysis with the right results. It will give the fault results if the instrument is not calibrated, or there is no SOP, there is no uh, guidelines are provided. So that's why the GLP is required for the accurate analysis in the lab, by providing the rules and regulations, so that we get the results which is 100% accurate, repeated, or repeatability of the results can be done, reproducibility, high quality results. So for all these things the good laboratory practices are required. So what is the definition of good laboratory practices? So GLP embeds a set of principles that provides a framework within which laboratory studies are planned, performed, monitored, and achieved and reported. So in simple words, they are preparing the guidelines, they are setting the rules, they are setting the regulations, they are providing the all the facilities, the infrastructure, the proper working condition in the lab, and the studies they are planned, they are continuously monitored, achieved, and reported. So it called as good laboratory practices. Are you getting my point? What is good laboratory practices? If you are working in a lab, and if you are provided with all the things, that is calibrated instrument, SOP, safety and hazardous conditions, all the laboratories are well planned, all the works are well planned, they are continuously monitoring, they are reporting continuously. So at that time, what the results you are getting, that results are 100% accurate, or it will give the best results. So that is the definition of GLP. So GLP is the set of principles that provide a framework, within which framework means they are providing the guidelines, they are setting the guidelines, the rules and the regulations. In the GLP, they are providing the facility, they are providing the infrastructure in the lab, which are properly planned, they are performed, monitored, and achieved and reported. So all these things it is called as GLP, that is the good laboratory practices. So next part, why this GLP was created. So in early 70s, the FDA they became to know that, or they became aware that there are some cases where the poor laboratory practices are going in all over the United States. Okay, so they discovered lot of faulty activities or frauds or the manipulation in the results or the poor lab practices. Some of the examples of these that they have visited some industries like in QC department they found that the equipment are not calibrated to the standard form. They are not well maintained. Maintenance is not done properly. And at that case they are found that they are getting the fraud results. That is the fraud activities are going on in the industry. So due to that they found that it is necessary to produce some good practices so that this kind of fraud activities can be stopped to get the poor accurate results. And for that, the FDA in 1978 they started the GLP or they create the GLP. That means we can prepare some rules and regulations, we can provide the infrastructure, we can provide the facilities to the lab so that it can maintain the safety of the person working in that lab, as well as it will give the highly accurate results, reproducible results. So due to these conditions, it is they found poor lab practices all over the USA. They have created these GLPs. So what is the purpose of GLP? So main purpose of GLP is that GLP is to certify that every step of the analysis is valid or not, and it makes sure the quality and integrity of data submitted to FDA in support of safety of regulated procedures or the regulated products. And third one, that is the third purpose of GLP is to have heavy emphasis on data record and specimen retention. So these are the three purposes of GLP, or these are the important purposes of GLP. First one, GLP is certify that what are the steps taken during the analysis is valid or not. They they assure the quality and the integrity of data submitted to FDA in support of the safety of regulated products. And third purpose of GLP is that it is making the heavy emphasis on the recording of data and the specimen retention. So these are the purposes of GLPs. So once again, I go through the introductory part of the GLP. So GLP is the Good Laboratory Practices. It is regulated by, or these regulations is created by the US FDA in the year 1976 and designated as new part of Chapter 21 of the Code of Federal Regulation, that is the CFR, that is 21 CFR, Part 58 in 1979. So why they created? So because they found that in the US, all over the United States in the lab, the false practices were going on. The instrument was calibrated. They are not performing the good practices. They are performing the poor practices or the poor lab practices. So to avoid all these things, they have included the good laboratory practices in the CFR, that is the 21 CFR. The quality system concerned with the organization process and the condition under the which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, archived, and reported. It is called as GLP. So what is GLP? A quality system concerned with the organization process as well as the condition under the non-clinical health. So we are seeing that laboratories, they are the non-clinical laboratories. Okay, so perform monitor, recorded, archive, and reported. Now we are on to the next part, that is the advantages of GLP. So in GLP we are following the good laboratory practices. In that we are providing with the all the facilities, the trained staff, the calibrated instrument, all the requirements what are there in GLP, all these are provided in the GLP or the good laboratory practices. Okay, so what are the results what we are getting? That results are accurate. So what is the first advantage of GLP is that it assures that the data are true reflections and result obtained from studies. And then it also maintains the safety, that is the preclinical safety and the residual safety. And if we are following the good laboratory practices, so we are generating the quality of data which having the high quality as well as the reliable test data. It is reproducible, reliable, high quality data. That is the third advantage. Then mutual acceptance of data, increase public confidence, and shorten the time to market for the new products. So these are the advantages of GLP. So GLP मुळे आपल्याला काय होतं, तर GLP मध्ये आपण सगळ्या फॅसिलिटी मिळालेल्या असतात, इन्फ्रास्ट्रक्चर सगळं व्यवस्थित असतं, सेफ्टीची काळजी घेतलेली असते, हजार्ड्स काय होऊ नये याची काळजी घेतलेली असते. त्यानंतर जे मेंटेन इक्विपमेंट असतात ते दिलेले असतात. दे आर वेल calibrated. The workers which are working on the analysis, they are well trained. So due to all these things, we are getting the correct data. We are getting the mutual acceptance of data. It increases the quality, reliable test data we are getting. So all these are the advantages of GLP or the good laboratory practices. So what is the disadvantages of GLP? If we are to perform GLP, we require more manpower. If we have to follow the rules and regulation of GLP, if we have to follow the guidelines of GLP, we require the number of persons more. And as the requirement of number of persons more, it increases the cost. So due to that, and we have provided all the sophisticated instrument and the infrastructure and the facilities, all including these one, it will give the increases the cost of the process. So the process becomes more expensive. And this is time consuming process. Okay, I have seen some students, they are taking the weight of the raw material of the reagents or the reactants during the lab. How they are taking, I have observed some students, they are keeping the butter paper, they are tear it, then they are adding the reactant. Suppose paramonobenzoic acid, they are adding the paramonobenzoic acid. Then the window which is present, they lock that window of the other door of the weighing machine, and then they are taking the weight. That is the actual SOP. Okay, but what the some students they are doing, they are only keeping the butter paper, they are not tearing, they are adding the sample or they are adding the reagent, then they are weighing, they are not at all bothering about closing the door of the weighing machine. It will also influence or it will affect the weigh of that sample. Okay, so that is the simple example. So it is time consuming or it will take more time. Okay, so that is the main disadvantage of GLP. If you have to go for the GLP, if you have to follow the GLP, it is time consuming, it is expensive, and it requires more manpower. The next part, what are the objectives of GLP? First one, GLP makes sure that the data submitted is accurate and it is the true reflection of the studies what we are actually doing. These the high quality results are produced. Then sure that data is traceable. And third one, it promotes international acceptance of test. So these are some objectives of GLP. Just go through this part, that is the GLP principle. Just go through it. What are you requires for the GLP to follow the GLP principles? So you should have testing facility. You should have that number of manpower for the testing. You have been storage of the records and the materials. You should follow the quality assurance program. You have to maintain the proper apparatus, material, reagents. Then you have to follow the test system. You have to prepare the test system. Then you have to take the test as well as the reference substances. Then the standard operating procedure SOP you have to maintain. Then facilities you have to be provided. Then you have to performance of your study and reporting of the study results. So all these parts it comes under the GLP principle. Okay, so these are the parts which comes under the GLP principle. Until now, it is clear what is GLP. In short, the facilities infrastructure that are provided for the proper maintaining of record to get the results which are highly accurate, reproducible. All these comes under the good laboratory practices. It also includes the safety of a person working in the lab. Okay, so then we have seen some principles, objectives of GLP. Now the principle next part, how to practice the GLP. So these are the 8 things through which you can practice the good laboratory practices. One, general provision. Second one, organization and personnel. Then equipment. Then the testing facilities, operations. Then test and control articles. Protocol for the conduction of the test and records and reports. These are the 8 parameters which comes under the GLP or good laboratory practices. So general provision means it includes the conducting of non-clinical trial studies that support the research and marketing permits of the products. It comes under the general provision. Then the organization and personnel means organization providing all the facilities, all the equipments, the testing facilities are they providing, or the personnel they are providing which are highly trained persons. The training has been undergone, or the person has been undergone the training. Then they are providing the testing facility operations. They are providing the SOP for the test, their control articles. They are providing the protocols. They are maintaining the records, reports, all these parts which comes under the good laboratory practices. Okay, so we will stop here. The details which are the parameters comes under the GLP we will discuss in next lecture. Okay, so I will once again go through what is the good laboratory practices. It is added by the FDA regulations. After seeing that in the US, all over the US, the false practices are going on, the poor lab practices are going on. They are not maintaining the proper calibration. They are not providing the facilities. They are not providing the trained persons. So by seeing all these things to maintain the proper quality of any testing or any material, they have introduced this part in the United States Food Administration. So this is the definition what we are seeing: the plan, perform, monitor, achieve and report it. Such laboratory studies which having some kind of framework It is called as GLP, that is the good laboratory practices. So it is why they are created. And these are the purpose of the GLP. These are the principles. Okay, so detail regarding all these things we will see in the next lecture.